In order to build probabilistic models of quantitative data, we have to model the quantitative data. That's the topic of this talk. Next slide. Quantitative data occurs with DNA evidence because PCR is a linear process. As we see in the picture on the right, the peak heights that we observe reflect the underlying DNA quantities. Therefore, we can use the quantitative peak heights and their stochastic variation to explain the observed data. The two-person mixture data with 3 peaks shown here on the right will be our running example. Next slide. Our basic genotype model is additive. It says that 1 plus 1 equals 2. That's different than threshold models that are all or none. Here, events can superimpose and add up the way we observe in nature. Let's assume the simplest possible mixture interpretation, where one of the 2 contributor genotypes, in this case the victim, at a 13-14, is known. And therefore the goal is just to infer one unknown genotype, that second genotype. Shown in blue is a candidate second genotype. In order to properly use Bayes' theorem to infer a valid probability distribution, you must consider all possible allele pair values and try out every candidate allele pair value. Let's start off with a very likely allele pair value, just to see what happens, and we'll choose the allele pair 12-14. Let's take from the first line twice the amount of allele pair 13-14 and add it to half the amount of allele pair 12-14. On the third line, we visually see the genotype pattern formed from that sum. There's an increasing amount of DNA at each peak as we move from 12 to 13 to 14. So this is a model. The genotype is predicting what we're going to see in the data. Next slide. The next step is to compare the predicted genotype pattern model that we're proposing to the observed experimental data. Remember, in Bayesian inference, you're not allowed, when inferring a posterior probability distribution, to ever touch the data in any way, but you must infer all possible patterns that your variables permit you. When we make this comparison, we see, looking at each of the 3 peaks, that we can line up allele 12, both proposed and observed, allele 13, the taller peak, proposed and observed data, and at allele 14, the genotype proposed pattern peak with the observed data. Next slide. To make a quantitative probability comparison between the predicted pattern and the observed data, we need a likelihood function. So what we do is at each peak, we look at the deviation between the height of the predicted peak and the observed data peak. We see that for allele 12, the peak heights are about the same, so that deviation is small. Similarly, at peaks 13 and 14, the pattern fits the data very well and explains it very well. And so again, the deviations at each of those individual peaks is small. The sort of likelihood functions that are simplest to understand are the ones where a good prediction gives a small deviation, and that gives a high likelihood. Think of a bell curve where you're measuring the deviation. Centered at 0 deviation is the top of the bell curve. And as the deviation increases by being off by too much or too little, you move down the sides of the bell curve until finally large deviations give you virtually no probability at all. That's the sort of likelihood function that is used in these problems. And so now let's move from the deviation to the likelihood. With a small deviation, we see there's a large likelihood. You're at the top of the bell curve near the center. That's true at peak 12 and peak 13 and at peak 14. So the likelihoods, how well the genotype pattern explains the data at each peak, is large in each case, we can multiply those large numbers from the top of a bell curve times each other and arrive at a product that's a large likelihood. Therefore, we'd conclude that the allele pair 1213 is very likely. Now, notice that what we're doing is we're combining the evidence. We're looking at each peak, and we're multiplying the likelihood at each peak. This, down on the bottom left in purple, you see that pi symbol, which means product in mathematics, where we multiply together the likelihoods, the probability of the observed data peak height, given that the question genotype variable equals little x, the particular unknown value, in this case 1214. And we multiply together these conditionally independent peak likelihoods in order to form a valid joint likelihood function. Next slide. Let's now consider an alternative value for the genotype. By Bayes' theorem, we must consider all possible different allele pair values, so let's try out a different candidate. Here, we'll try the second genotype pair, 1213, and we'll again consider the same mixing weight of 2 to 1. When we add it up, we get a new genotype pattern that we're going to propose as a prediction to compare against the data. And here we see the prediction for allele peaks 12, 13, and 14. Next slide. We must compare the model to the data, and we do this peak by peak, because by comparing the separate independent peak events, we'll be able to combine their probabilities. So, taking a look at the predicted genotype pattern allele peak 12, we see that it matches up very well with the data. However, there's a peak height mismatch at 13. The proposed peak is too high to the observed peak at 13, and at proposed peak pattern 14, where it's too low. The peak height is too low at peak 14 of what's proposed relative to the observed data. Next slide. We now form the same sort of likelihood function, where we examine quantitatively the deviation between the predicted peaks on the left and the observed data peaks on the right. We see that at allele peak 12, indeed, the deviation is still small. However, at allele peak 13, there's a larger discrepancy. The deviation is higher, and the same is true at allele peak 14. So what we're predicting, what we're proposing from the second genotype candidate 1213 does not produce a pattern that fits the data very well. This is expressed in the next line, where we qualitatively show what the likelihood would be. There's a large likelihood for the small deviation at allele peak 12. However, as you move off the top of the bell curve with larger deviations and you move away from the center, those larger deviations produce much smaller likelihoods. And so at allele peaks 13 and 14, there's now a small likelihood from those large deviations. When you multiply together using the product rule of independent probability events, the large likelihood times the two smaller likelihoods at the other two peaks, we arrive at a smaller likelihood than we had before. So the allele pair 1213 is less likely than the preceding candidate allele pair 1214. Here again, we're making use of a joint likelihood function, being able to multiply those independent likelihoods at each peak to form the composite joint likelihood function across the entire locus. Next slide. Enumerating all genotype possibilities in this way, and in practice you would do it for all possible mixture weights as well, as we'll soon see, we can list for each allele pair what its likelihood is. For the 1214, it had a large likelihood. Suppose we would look at it and say, oh, it's about 90%. And then for allele pairs 1212, 1213, as we just saw, it's small. Maybe we'd give it a 5%. Now, other candidates don't even explain the peaks that we're seeing in the data. For example, the 1313 allele pair doesn't even account for the 12 allele that we observe. And so a few other candidates may get a very small likelihood, 1313, 1314, 1414, while the other allele pairs that contain alleles that aren't even included in the observed allele peaks would obtain essentially no likelihood at all. Moving on to the last column for the probability, we have to take these values, like small, large, whatever the numbers would be, and renormalize them to add up to 100% to get a probability distribution. Of course, to get a well-founded probability distribution, the likelihoods must be multiplied by the prior in order to form an appropriate posterior distribution using Bayes' theorem. This is the sort of probability distribution that we see when we hand out this quantitative data as an exercise to forensic scientists to interpret using this method. Interestingly, the inclusion method would give all of the first 6 allele pairs on this list equal likelihood and hence equal probability, which is not what people actually do when they have the quantitative data. Even if you used an obligate allele method and said, suppose that 12 must be an allele, then you'd have just the first 3 allele pairs on the list, and you'd have 1/3 probability using that method. That's what the current interpretation methods that use thresholds currently assign, whereas when experienced DNA analysts look at the data and walk through exercises like this, they tend to assign far more probability to the more probable allele pairs. Next slide. Genotype inference from mixture or any other DNA data is done by every known method using Bayes' theorem. The posterior probability distribution of a question genotype at each allele pair value, the probability that capital Q equals X, conditioned on the observed data, must be proportional to the likelihood, that is, how well the hypothesis that Q equals X of that particular genotype value explains the data, times our prior belief, our initial probability based on the population, the probability that Q equals X. By trying out all possible values for the questioned genotype random variable, capital Q, tend to give higher likelihoods that better explain the data, and thereby, when combined with priors, give us more posterior probability. Here again, if you see in the bottom left, that's a product of likelihoods. I've indicated that it's the data at each locus. We've already seen that we can combine the peaks at each locus to form a likelihood function for that locus. But by the same principle of independent peaks and independent data, we can form joint likelihood functions that combine the information across multiple loci. And that's the same joint likelihood function. Next slide. Let's see how these patterns apply to data. Recall that peaks are probabilities, and therefore when we consider data uncertainty, we don't have one value of a peak, but it's a representative of a probability distribution shown by the black probability distribution in monochrome on which a genotype probability pattern is overlain. When, as in this roughly 2-to-1 mixture, we see that the predicted peaks lie where the data are in probability, then we can account for the data uncertainty, those stochastic effects, and the proposed allele pairs that form these patterns, shown by the heterozygote in blue at a lower amount and the homozygote in orange at a higher amount, will get a higher joint probability. However, next slide, when we propose genotypes of alternative allele pair values that do not coincide with the observed peak data, then we'll get much lower likelihoods. Here, for example, we've moved the small allele on the left in blue over to the right, and now there is no longer any pattern associated with that first peak at around 75 anywhere. So that would have a very, very low likelihood. Moreover, where we've moved that little blue allele to, it's predicting an event that's not occurring. And so there again, it would have a low likelihood. So in this way, by proposing all possible genotypes and generating all possible allele pairs And comparing them with the probability distribution of the observed data, we can determine which genotype values are more probable. Next slide. Bayesian probability has us assess all possible genotype values and the patterns that they produce, comparing them with the data in order to determine the probability of each allele pair. As we're about to see, you can similarly compute any random variable, including the data variance. So when you determine computationally that there's a small amount of data variation, it means there's a low coefficient of variation, and the probability around each peak is tightly clustered around the observed peak data. That gives a very restrictive pattern that only a few genotype patterns and their underlying values can possibly match. And therefore, we end up with more certain genotypes. Very few possible patterns, therefore more certainty in the patterns that we do have. However, if the data indicate that there's a large amount of variation, that becomes permissive. Many possible genotype values become possible because the distribution of the peak expands to allow very many different patterns and therefore very many different allele pairs. By diffusing the probability across these very many different allele pairs, we have less certainty. So uncertainty in our data translates into uncertainty in our genotype. Next slide. And that in turn translates into uncertainty in our match statistic. So as you recall, the likelihood ratio match statistic reflects the genotype of uncertainty that we observe by moving us to a posterior genotype probability in the numerator relative to what our initial belief was about the genotype value in the denominator. The genotype certainty concentrates probability on just a few good bets and focuses the LR on what's more probable, yielding more information. However, when the genotype uncertainty diffuses its probability because of uncertain data across many possible allele pair candidates, that will reduce the numerator here. It will reduce that concentration of probability at the event that random variable Q equals the suspect's genotype S conditioned on the data, and by reducing the ratio, reduce the likelihood ratio and give us less identification information. Next slide. Exactly the same Bayesian principles apply to mixture weight inference, as they must. The posterior distribution of a mixture weight value between 0 and 1 is proportional to a joint likelihood function times a prior probability. Here, the joint likelihood function is how well a particular mixture weight explains the observed data in the context of all the other variables, such as the genotype allele pairs, stutter, relative amplification, uncertainty of the peaks, and so on, multiplied by the prior. We want to consider the data at all loci, and so as we see in the bottom left in purple, there's a joint likelihood function that's the product of the likelihoods of what happens at every locus. We must try out all the possible mixture weight values between 0 and 1, or if there are more than 2 contributors, different pairs of numbers, triples, quadruples, whatever needed, that will add up to 1, in order to completely explore the space. But when we're done, we'll be able to determine the posterior probability distribution of mixture weight. Let's look at this visually. Next slide. We'll take a look at the mixture weight probabilities accounting for the underlying data peak uncertainty. On the same graph that we've seen of different STR peak sizes and RFU on the left, we have different STR peak probability distributions shown in monochrome. On top of that is a pattern formed by assuming the 2 alleles for the first contributor in blue that are low, and assuming the homozygote allele pair shown in orange for the second contributor. When these are put together and compared against the data, we see that this roughly 2-to-1 mixture weight has a good fit to the observed peaks within those probability distributions. They're in the region as determined by how well they're hitting the shades of gray. However, next slide, when we consider an alternative mixture weight that's flipped, it's say 1 to 2 the other way around, now the predicted pattern of the first contributor alleles in blue and of the second contributor allele in orange are no longer landing where the data are. They're not in the region of probability of the observed data peak, and therefore this mixture weight alternative would have a very low likelihood, leading to a low probability. Next slide. We can do exactly the same thing with data variance. If you recall from the bell curve, the normal distribution has an exponent that includes the data, the mean, and the variance. Well, the data is always fixed in Bayesian statistical inference, but the mean and the variance are parameters to be determined up to probability. To determine the variance, we just use Bayes' theorem again. The posterior probability of the variance random variable, capital V, is having some value, little v, conditioned on the data, is proportional to a joint likelihood function across all the different data peaks times a prior probability of what the variance would be as a probability distribution in the STR experiment. Again, we have to try out all possible variance values, typically by statistical computer simulation, and then examine the joint likelihood function, which we see on the bottom left, which combines the evidence in a statistically valid way across all the data peaks, and when it's combined with the prior, will give us a posterior probability distribution as to what our certainty in our data peaks is. Next slide. Suppose now that our focus is no longer on the genotype allele pair values or on the mixture weight values. Instead, what we want to understand is the data variance, the probability of data peak uncertainty. From first principles, we know that as we have taller peaks, there's a larger absolute standard deviation, but it's more concentrated on a relative scale. There's a smaller coefficient of variation. And we can therefore model our data variance as the height of the peak times a scaling factor. We're seeing here a relatively typical scaling factor for STR data, and we see a good correspondence between the color pattern that's being predicted against the variance of the data peaks. However, next slide, when that scaling factor increases, the data variance probability distributions around each peaks expands as we see its larger absolute standard deviation for the taller peaks, lower at the smaller peaks, but this is an alternative value that mathematically does not explain the observed data and pattern interaction as well. And therefore, this larger data variance would end up with a lower likelihood. When trying out all possible values, we'd end up with a probability distribution centered on more peak certainty, as we saw in the preceding slide, rather than more uncertainty for the data variance as we see in this slide. Next slide. In conclusion, quantitative data modeling is a standard way to proceed in science. You identify what the different variables are as random quantities and how they interact, how the patterns that they form can be compared against data in order to form likelihoods that would explain the data. Using Bayes' theorem, combining with priors, you arrive at posterior probability distributions regardless of what the variable is. It's the same rule. There is only one rule of valid probability reasoning, and that's Bayes' theorem. Here, the genotype for STR typing is the main variable of interest, and that's because it's only the genotype that can give us the identification information that we need for the likelihood ratio. Mixture weight is useful to people and to the computer as an explanatory variable. as well as the data variance or stochastic effects that can be calculated in order to determine how certain our answers are and end up translating into certainty of genotypes and likelihood ratios. To preserve all the identification information present in the original quantitative data, quantitative probability modeling is required. Thank you.